Thank you. All that to just move on to get some circles. So what I wanted to point out was this year we have a mismatch in the vaccine strain and the circulating strain. Few amino acid differences between those two, and we have very low efficacy. Depending on the different reports you see, it's either no efficacy or 15% European, Canadian, U.S. reports. But essentially, it's the vaccine hasn't been working this year. But what I'd like to go to is it's not just that it's not that it is mismatched, that this study was referenced last year, that even on a good year, the efficacy of the current vaccines is at best 60%. So this CIDRAP report is very comprehensive and was solicited after the 2009 pandemic response and evaluated all the studies from 1967 on and eliminated all the studies that didn't meet the stringent requirements for endpoints and for study conduct and all that. Because there's this misconception that the vaccine is fine, and that's been cited in this report as a barrier to novel vaccine development, that if people think that it's only when there's a mismatch that there's a problem, that really it's just fixing how to keep up with the evolution of the virus. So there hasn't, at best in adults 18 to 64, 60% efficacy. In the age group 2 to 17 years, inconsistent evidence of any protection. And for the elderly, no evidence. So flu vaccine efficacy must improve. And it's not that there isn't a commercial need. There is a big market, and we do have representatives of big pharma here who could speak to it better than me. And when you look at this flu vaccine, the efficacy is much, much lower than what we accept for all other vaccines. It's 90% or better for all the traditional vaccines that we do use. It's only flu that operates in this 20 to 40% efficacy, and that we accept as the status quo. And influenza annual cost, you can measure it from life years lost to hospitalization to medical costs, economic burden. It is high, it is there. And the deaths, the population that's most affected is the elderly. They're also the most compliant group that take the vaccination, right? It's either the very young or the elderly that are compliant with flu vaccine usage. So the current flu vaccines simply don't work well. We said that the split vaccines just rely on the HA antibody, and that the other vaccine that's on the market, the live vaccine, suffers from pre-existing immunity in the adults and is efficacious in the children. Now, flu vaccines have been on the market for last, 40 years, and only recently have we had new things approved, the new flu block, flu cell vax. A lot of these are to keep up with that pandemic manufacturing need to not rely on eggs and all, and only the Fluzone high dose is the one that's been targeting a population, the elderly, and increasing the dose of the HA. But again, all of these are variations on the same theme of relying on the HA antibody response. In nature, we do know that potent immunity develops after a natural infection, that all other parameters besides HA antibody does provide protection. This has been shown in the different antigenic shifts in the pandemics we've had in 1957, 68, even that 76 and 2009, that there are other correlates that play a role and mitigate the disease. 2009, T cells were shown to provide protection. And then there are other parameters like the mucosal IgA that we discussed today, and I'm glad that there's more progress being made on that because it's been in the literature. John Trainer has always shown that in collaboration with the IgG, the nasal IgA does contribute to protection. MedImmune's Flumis in the insert mechanism is not known, but T cells and IgA mucosal antibodies do contribute to the protection observed. But as discussed earlier, the mechanism of how do you measure these assays and things aren't available. We always rely on this HAI antibody, and that's one of the other things that's been cited at the CIDRAP report, that this is the path to licensure, having an HAI antibody response. So anything that diverges from this, it's hard to go on. So what defines universal then? We've heard talks today about broad HA coverage, so universal meaning within a subtype covering all the different strains. Universal meaning the immune response covering T cells and mucosal immunity. And then these other new targets like the HA2 stem and the M2E antibodies. And universal, is it like polio? You get it once and one dose for multiple seasons or forever. And I think for flu, given that the HA mutation rate is what drives the vaccine, it would be hard to achieve that one dose forever, but the others in combination may be possible. So what would a better flu vaccine look like? And I don't know if all of you may be aware that BARDA has issued an RFP for universal influenza vaccines, just like BARDA drove probably the manufacturing one. The government will be driving the next probably what the universal vaccine is going to look like. And the key potential attributes from this RFP, the target product profile, did list protects from multiple flu A subtypes and most B strains, superior efficacy in elderly, duration, 1 year plus duration of immunity, and one dose for all ages. I think, Josh, you talked about duration, but one of the problems with the current vaccines is that it wanes after the response wanes within 3-4 months after immunization. So that also is a target in our trying to design better vaccines. And these are, maybe you've seen press releases or heard all of these, but these are the new universal vaccine players. They all rely on one component. The approaches are adjuvants. We've heard some here at this conference, more live attenuated and vector-based programs, and T-cell only based vaccines, whether it's peptides or a string of epitopes, and then other systems for VLPs, HA stem, or RNA or synthetic nanoparticle type things. But all of them, when you put together the competing technologies and how do you get to licensure, the things that facilitate technologies that resemble the current technologies that that are known, have a regulatory path, elicit HAI, those are the ones that are closer to licensure. The others do need more definitions and more things developed for it. Yesterday I spoke about FluGen's M2SR vaccine. We fit into the live single replication category. And we have universal characteristics, cross-protection, HA2 antibodies, and intranasal delivery. And it goes through all these different responses, but only the HAI is the one that really goes to licensure. So our target is as well to call it a seasonal vaccine, because with HAI, if you do get your seasonal match, you can go through the licensure path, but we want to say it has universal attributes. We have those other T cells and other responses. Okay, this is So in phase 1, we're going to be looking at all these other responses that we observed in animals, but only the HAI antibody really has any regulatory weight. What do we do with the other ones? Essentially, we're going to measure in a first in man, but if no HAI responses or your HAI responses don't meet the licensure criteria, you're going to have to establish your own correlate. And the best way is to start using challenge models, human challenge models. We heard earlier today on the influenza challenge studies, they are being used more and more, and there are now more service providers that do these. But as Rob said earlier, it's not going to replace the trial or an efficacy trial, but it will give you proof of concept, and FDA is looking more and more at it, and one strain of Flumis was approved on this. So in summary, the types of things that I guess we're all encountering are the establishing the appropriate correlates of protection, the assays that are needed when your technology platform differs from the current model, and the challenge models that help to be able to define things associated with them. So I think from here we can go on to any of our panelists that are sitting in the front row or here to discussing what types of issues people have had with their technologies. And I'm trying to escape this, but I can't.
That's the technology you're having problems with.
Okay, thanks. So do we have any other presentations, or did you want to talk about your antigen development to match your bacterial things that you can't use the SRID, or?
I think we pretty well covered that in the talk yesterday, but just throw that out as something for people to think about with regard to novel technology that, if the SRID is not working, looking at alternatives. There are some publications on, for example, a Biacore SPR type measurement of sialic acid binding, which is it's nice because it's independent of any neutralizing antibodies, so you don't have to wait for the reference serum to become available. There are mass spec methods using isotopically labeled peptides as reference antigens, so you can quantitate that way. And then there are attempts to use sort of broadly neutralizing monoclonals as reagents, either in ELISA or perhaps a chip type format to quantitate. So again, you don't have to wait for the strain-specific reference serum to come out for that particular HA.
I guess, one thing, again, there's numerous difficulties, especially when you start getting into the universal question. One thing that stands out in my mind with anything that's live at any point in its life cycle, in other words, the IIV as well, I think yours might have the same issue, is that we already know that culture adaptation mutations can be a problem for the current seasonal vaccine. We can get just mutations that affect your immunogenicity. Now, if you're talking about in the context, and you can try to control for that to some degree to look at the antigenicity or immunogenicity of the currently circulating strain. How do you deal with that in the context of a universal that you're claiming to protect against anything that comes out? If you get a mutation near the receptor binding site and you're still reactive with the currently circulating strain, are you really confident that you're still universal? Right. That is, with anything live or replicating that's going to pick up mutations, a key consideration, and it's a challenge.
Yeah, and how would you get that label and maintain it, right? What does that mean?
The label, that's a whole other question.
Three years of three seasons, 4 seasons.
And that gets to your earlier question about what is universal, what is breadth. I mean, H3 provides a bit of a better opportunity to establish some proof of universality because we have had recent, we have, first of all, it's more dynamic overall, and we have had a recent shift. So, you know, anything you had up to that point, if you're claiming it's universal, you better get that shift. H1 is more of a challenge. It's been sitting around for 5 years. But, you know, how do you, again, define universality? How do you use old strains to say you're going to protect against the future? I think that's a challenge we're all struggling with. What are we going to do to define universality? So, and even when you say, yes, I'm inducing responses against numerous strains, is it as good as the matched HA? Because that's your competitor. As soon as the shift happens, your competitor is a well-matched HA. And just because you're better for the half a year or so, because you have some breath, is it really going to outcompete the matched HA that next year, which could be your competitor? I think these are all questions that come into play with the universal. So.
Is there any questions from?
I've got a question, and it's like if you wanted to do some sort of blue skies, if you wanted to do some sort of blue skies thinking about this, let's be honest, using the HAI assay, because that's what the World Health Organization wants to do. And bearing in mind my PhD was based on it, as I said earlier, it's like, you know, so I'm not going to criticize it too much. But for efficacy, what would be the other way to actually, because I think it's flawed using the HAI assay. So what other assays would we like to use?
So now there's more of a shift to microneutralization, right, as a measure. Because what does HAI mean? We say 1 to 40, but it's what, 50%?
Yeah, but it's, yeah, absolutely. So.
And I think the high-dose trials showed, you know, even though they had twofold increase in HAI titers, efficacy moved 24%.
I think everybody has given up on the, and no disrespect, but the Hobson paper is pretty much sidelined now. It's the 1 in 40, you know, it's just what they came up with then, and that's it. So we've moved on from that now.
So I agree, largely. That being said, the recent high-dose study, I hope I'm allowed to say this. So I recently was at a
Turn off any.
presentation. Yeah.